All right, well, it's a pleasure to be here and talk to you about some technology that my company's been developing to deal with stress tolerance in plants. And the motivation for our developments are based on a lot of things we're seeing happening around the world. Many aspects of climate change are resulting in decreased water availability and quality as well. And so we began our studies by simply asking, with these stresses coming on, how do plants and natural ecosystems adapt to these stresses? Because there are stressful environments all around the world. We are interested in sustaining agriculture in the century and looking at these plants in different habitats that impose stresses that require adaptation by the plants, we thought would give us a clue to this. And it's kind of interesting because as you go to higher and higher stress habitats, the number of plant species that you find in those habitats decline. So there's an inverse relationship between stress and biodiversity. What's interesting to me is that in all of these habitats, sorry, let me start over there. With all plants have the ability to perceive stress and respond to that stress. But very few actually have the ability to survive the stress. So although they all kind of do the same thing with regard to perceiving and responding, very few can mitigate the impacts of that stress and survive. And so we looked at a lot of the literature over the last 100 years to see why this was and what the understanding of adaptation was. And there's been, oh gosh, more than 100 years of research by a lot of very intelligent people that concluded that organisms adapt to stress by making modifications in their own genomes. So they're confronted with a stress, they modify the genes in their own genomes that slowly allow them to adapt to those stresses. That's basically Darwinian evolution. And that spurred two developments for agriculture in an attempt to generate stress-tolerant plants. One is breeding traits from plants in high-stress habitats. The other is genetic modification in one form or another, either transferring genes through new genetic technologies or by doing selection, by mutational selection of plants. But one way or another, it's either genetic modification or breeding. The problem is that there hasn't been much success along these lines. So there have been incremental increases in stress tolerance in crop plants, but in reality, there's very few shining stars that indicate that either one of these techniques have been working well. The problem really comes down, and where we come into the whole scene, is that all plants and animals on this planet are symbiotic with microorganisms. It's an inextricable association that occurs in a fundamental component of life on this planet. Humans have more bacterial cells than human cells in them. And without those bacterial cells, human health plummets, and actually for the tests that have been done, the studies that have been done, survival is greatly reduced. We are wholly dependent on those organisms for nutrition and health, as are plants. So both plants and animals share a lot of commonalities with regard to symbiosis in that, one, they require them, two, they both associate with bacteria. However, plants are a little bit different in that they also associate with some other groups of symbiotic organisms. That we know that there's viruses involved in these plant associations. We know that mycorrhizal fungi are associated with plants as well. Those associate with roots, grow into the rhizosphere, the root soil area, and act as an extension of root systems. So they can transport nutrients into the plant and they get carbon back. As a matter of fact, a lot of the things we eat are mycorrhizal fungi, the fungi that we all love to put in our food. The other group, and the group that I'm going to talk about today, are known as endophytic fungi. And these are fungi that live entirely inside the plant. And they only come out when the plant dies. And then they'll sporulate on the surface, and the spores will drop down in the soil. But otherwise, they reside entirely inside the plant throughout the life of it. Now, there are 4 groups of these endophytic fungi. And let's see if I can get this going. If any of you are familiar with endophytes, you're probably familiar with this first class 1 group. They're associated with cool season grasses, and they've been a problem in forage grasses for quite some time because they make these really toxic alkaloid chemicals that impact animals. The group I'm going to talk about today are these class 2 endophytes, where the class 1s have a narrow host range. They're kind of restricted to these cool season grasses. The class 2 endophytes have very broad host ranges, and I'll show you the impacts of that. They also have some very unique fitness benefits that they confer to plants in the form of stress tolerance. So what I'm going to do is I'm going to take you through the development of our first product, which we call BioEnsure. That is a seed treatment, and I want to show you the utility of this and the impact of the seed treatment on plants. But I want to take you through how we got there first. And our studies began in Yellowstone National Park looking at these geothermal soils. These soils get down to about 20 degrees centigrade in the winter, and they get up in the 65 to 70 degrees centigrade in the summer. And there's a small community of about 8 plant species that live in these soils. We focused on one plant species, which is this tropical panic grass that grows in the hottest soils and grows isolated from the other plants in that community because the other plants can't get to that high a temperature. And what we found was that all of the plants we looked at a couple hundred individual plants, they were all colonized by one fungal endophyte. And this turns out to be a class 2 endophyte that's Curvularia protuberata, and it grows in all parts of the plant except for the seed. So that allowed us then to bring plants back to the lab, take off the seed coat, which had the fungus, and then we could grow up the sterile seed from the fungus, which we could also culture, but we could grow them separately from one another. And we could ask very specific questions about the importance of the association to both the plant and the fungus. And the first thing we found was that neither one of these symbiotic partners could grow above 38 degrees C when grown in isolation. But when we put them together, or certainly we knew in the field, they could grow up to 65 or higher. And so we set up some experimental conditions where we could heat the soil of the root zone to whatever temperatures we wanted to, and then we could compare plants where we put the fungus in, these are symbiotic plants, to plants where the fungus was absent or non-symbiotic plants. And then we cycled these plants at 70 degrees centigrade throughout the day. We turned them down to 37 at night, tried to simulate the diurnal cycle we see in Yellowstone. What we saw was that within just a couple of days, the non-symbiotic plants all died. Some of them died so fast that they didn't even have their chlorophyll bleach out. They dried up and just croaked. Whereas the symbiotic plants survived until we terminated the experiment. In this case, this was 10 days. Well, it's fine to do experiments in the lab, and I can tell you right now that if we looked at the literature and transferred everything from the literature into agriculture, none of you would be complaining about any problem, period. There'd be no insect problems, there'd be no stress problems, there would be no problems. But the problem is that what you see in the lab doesn't necessarily always translate out to the field. And so in all of the ecological studies that we've done and the agricultural studies we've done, we always start in the lab, but we always transition out to the field to see if there's any reality to the laboratory results. In this case, what we did is we backpacked out 1,000 pounds of soil from our site in Yellowstone. We sterilized it, and then we carried it back in, put it in these tubs, and then planted either symbiotic plants or non-symbiotic plants. We did that over a temperature range, and what we saw was that when we hit 40 degrees in the soil, the non-symbiotic plants did not survive, but the symbiotic plants did just fine. And in fact, throughout that temperature gradient, the symbiotic plants were always bigger and healthier compared to the non-symbiotic plants. So this was a clear example, not only in the lab, but also in the field, that a symbiotic association between a plant and a fungus had a lot more to do than just nutrient exchange. In this case, they were providing survival to one another in the form of heat tolerance. Okay. So how did we get to BioEnsure, and why did we even develop a company around this? Well, it turns out that these fungi that we were isolating out of these plants have broad host ranges, as I mentioned earlier, and we can move them into agricultural plants as well. In this case, these are watermelon plants that are symbiotic with the Yellowstone fungus. These are non-symbiotic plants, and we put them over the same temperature range that we did for a lot of the Yellowstone plants as well. Well, it turns out that when we put the fungus inside the watermelon plants, they become heat tolerant as well. So normally, like many plants, they can only grow up to about 38 degrees centigrade. With the fungus, we can grow them easily at 50 degrees centigrade. So this functionality then could be moved from one plant to another. And for us, what the reason we chose watermelon as an example is because watermelon is a eudicot, and the fungus came out of a grass, which is a monocot. And those two plant lineages diverged about 200 million years ago. So the communication that's involved in this heat tolerance and survival of both partners is extremely well conserved. And it's the same thing in both of these plants. And that sort of then was the impetus to try this on as many plants as we could. And it turns out that these fungi have extremely broad host ranges. So we've tried them on a variety of cucurbits and solanaceous plants and chrysanthemum and fruits, and also what you call broad field crops as well, which is what we've been doing a lot with of late. Well, that led us into looking at the ubiquity of this association and this phenomenon. How common is it for plants that are adapting to stress to have an organism inside them that actually adapts them to that stress? In the Yellowstone scenario, those plants were not adapted to the stress, right? The fungus was adapting them. So you take the fungus out of the plant, and those plants that have been there for millennia can no longer exist in that habitat. Right? So we've looked in various habitats. There's a couple of examples here. And we now know that when we go into a habitat and we can identify what the major stressors are, that we know there's a fungus inside those plants conferring tolerance or adapting those plants to those stresses. So on the coastal beaches of Washington State, we started working with a grass here. All these grasses that were on the beaches that get inundated by high tides had a fungus inside of them that we could move around to different plants, and we could grow watermelons or rice at anywhere from 300 to 500 millimolar sodium chloride, which is similar to seawater. And I'll show you an example of that. We refer to this as habitat-adapted symbiosis, and we've looked at numerous habitats now, and we've been able to generate or demonstrate that the fungi out of the plants in these habitats that require adaptation to stress can be moved into these agricultural crops and confer the same functionality, whether it's to high-energy explosives like TNT or drought or heavy metals. And here's an example of tomatoes grown in, I believe this is 300 millimolar sodium chloride. This is without the fungus, this is with. In each panel, the non-symbiotic plants are on the right, the symbiotic plants are on the left. And so there appears to be no limits to what these symbioses can achieve. The only thing that we can conclude at this point is that when you go to high-stress habitats, very much against conventional views in ecology, adaptation, evolutionary biology, the plants themselves are not adapting to the habitats. They require a symbiotic association to adapt to that. So this has really big consequences in a lot of plant ecological studies, also climate change studies. Right now, none of the climate models, for example, take into consideration how plant communities will move. We don't know the distribution of these fungi. We don't know if, for example, if a place dries out, will there be a fungal endophyte available in that area that can colonize the plants and allow them to sustain themselves there or adapt to that new environment? These are still big questions in plant ecology. But with regard to agriculture, we have been developing these organisms to apply to seeds of virtually any crop and virtually any variety of any crop to confer the functionalities that we're interested in. We've gone to great lengths to try to determine how ubiquitous this phenomenon is. We've looked at plants from the Antarctic, we've looked at plants from the Arctic, and we were also able to get some mosses from almost 6,500 meters off of Mount Everest. It turns out that no matter where we go, these plants have these symbiotic fungi in them. They all have some functionality that confers benefits to those plants, allowing them to be in those habitats. Okay, so that's the stress side of things. There are some other components of these interactions that I wanted to show you as well, and the first of which deals with the impacts of BioEnsure on the growth and development of seedlings. Now, these panels are representative of many different plants we've worked with, The left side in each panel are symbiotic plants. The right side are non-symbiotic plants. These are the same age plants in each of these photographs. And you can see that the impact of the bioinsured organism can be fairly dramatic on the growth of the plants. Now, on top of just the basic growth, we see a tremendous impact on the development of plants as well. And I've got a couple of time-lapse videos I'm going to show you. These are rice seedlings that are either symbiotic on the right here or non-symbiotic on the left under two conditions, 24 hours of light or 16 hours of light. And let's see if I can get this happening. Now, what happens with this is that these symbiotic plants will extend these root systems about 5 times the speed, or they're growing at 5 times the speed of these non-symbiotic plants. More importantly, though, they are allocating their carbon preferentially to the roots until root hairs start to form. And once those root hairs come out, then they start putting a lot of money into their, or a lot of carbon into their shoot system. Right, so it's exactly what you want a plant to do. You want it to establish a root system, put root hairs out, be able to start extracting nutrients from the soil, and then start expanding its shoot system. Over here, what you see is that the carbon allocation is about equal. It puts about the same amount into the shoots as it does the roots, and of course, it doesn't get nearly as big. Now, this is, again, under 24 hours of light. When we switch to 16, the situation changes quite a bit. Now what we see, I've removed nighttime so it gets a little jerky, but the expansion of the roots, the symbiotic roots are the same here as they are over here. They grow at about 5 times, well, in this case it's much greater because these roots never start. The carbon allocation here is the same. They preferentially put carbon into their root systems until the root hairs start developing, and then they put it into their shoot system, just like we saw over here under 24 hours of light. The non-symbiotic plants here put all of their carbon into their shoots. So over an 8-day period, they never started their root systems going down. Now, in agriculture, we compensate with our various inputs we put into the soils, but this is the way it works in nature. The other component of this, and to me the more important component of it, is that in order to generate these, this is the same on both sides of this black thing. This is just a separator, and this is just an auger medium. So it's basically like pouring Jell-O down between 2 glass slides and then growing the plants through them. But after several attempts with the non-symbiotic plants of putting them in here, the roots couldn't penetrate the auger, right? It's like you not being able to put your tongue into Jell-O. It's just a very soft matrix. Instead, what would happen is the roots would start to grow and the entire seed would just grow up out of the assemblage and fall off. And so in order to do these, I had to actually poke holes in here to stick these roots into the auger so it would grow. So they lose, it's not just the allocation of carbon, it's beyond that. The strength and the ability of those roots to penetrate into substrates is diminished. Now, the other thing that BioInsure does is it confers drought tolerance to plants. And we do extremely sophisticated drought experiments in our lab. What we do is we grow up plants to a certain point and then we just stop watering them and asking them how long they live. So we basically look at time to wilt. And that's what these numbers are. So these numbers here are just days without water. You can see in wheat, even at 18 days without water, we still have live plants. These aren't going to probably produce very much yield, but certainly at 15, they're just fine. And same's true with tomato, 14-day wilt times with a fungus inside of it versus 6. These are ongoing experiments we have right now with corn and soybean, and these are the days of drought that they've been exposed to. The non-symbiotic plants are dead here, and the symbiotic plants retain all of their turgor at this point as well. And the way this happens is that when we look at fluid usage, so we're looking now at water consumed by these plants over a 5-day period. The blue bars are non-symbiotic plants, and the red bars are symbiotic plants. Sorry, it's a little bit of my jet lag, I think, getting involved in my confusion. But anyway, so when we look at the blue bars, we see they're always bigger than the red bars, which means that the non-symbiotic plants are consuming significantly more water, up to 50% more water than the symbiotic plants. Or let's put it the other way. The symbiotic plants are consuming up to 50% less water. Now, this does not translate into any metric that's measurable with a lichor, for example. So it's not that the symbiotic plants shut down their stomates. It's not that transpiration rates alter. It's not that photosynthetic rates change. Those are all the same. But they are literally taking up less water, yet retaining all their turgor and putting on more biomass. It's a bit of a conundrum. But what happens is that they become more metabolically efficient. So once we put these fungi inside these plants become a different physiological entity. Their gene expression patterns change, their protein profiles change, their behaviors completely alter. The other, and one way we have viewed this is looking at nitrogen limitation on these plants. Corn is known to be quite a hog of nitrogen, and so these are 3 varieties of corn that are non-symbiotic, the Yellowstone fungus inside them, and then our BioEnsure product, our latest product, and these are all grown for the same length of time on nitrogen-free soil. And these basically grow out for a little while, peter out. These guys can put on significantly more biomass as a result of that. So they use what inputs they get much more efficiently. And so, we're looking at a scenario of less water, more biomass, and less input. Okay, now a lot of the pictures I've shown you have been from greenhouse and laboratory studies. And so the question you should be asking is, fine, what happens out in the field? And so we've done a series of field testing on different endophytes over the years, and I just sort of synopsize it here, and I'll get to the graph here at the end. But we've looked at corn, wheat, rice, watermelons, and a variety of cucurbits. I didn't put the peppers or tomatoes in here. But basically, whether we have stress or not, the take-home message here is that in the presence or absence of stress, we see yield enhancement as a result of these symbiotic associations. As you transition from no stress, well, except for the cucurbits, as we transition from no stress into higher stress situations, we tend to see more impact of the association. So last year, we did some corn trials in the state of Michigan, and we just happened to have our plots go into the county that was the hardest hit by the drought in the US last year. And although there was a fair amount of deer herbivory, we were able to get a lot of information out of the plots, and what we saw was that the plants treated with BioEnsure, they produced as much as an 85% yield enhancement over the control plants. We got about 125 bushels per acre out of the BioEnsure plants, around 65 out of the control plants. And in this area, the production on a normal year is around 140 bushels per acre. So we were quite delighted with the outcome of these experiments. And the most important thing I want to point out here is that under all the conditions we've looked at, under all the different field trials we've looked at, we've never seen a negative response. Now, I'm going to finish with this slide. And this is a study we've done in cooperation with a South Korean group. And the South Koreans had gone out and isolated a large array of weedy rice plants from 2 different habitats. They got 2,000 isolations. Half of them were from a drought stressed area, half of them were from a salt stressed area, and they brought these in. They did a lot of testing. They showed fantastic stress tolerance in these plants, and then they started a breeding program to move the traits of these wild lines into their favorite commercial variety, Dongjin rice. Well, they sent us a number of these lines, and when we looked at them, all of the drought stressed plants had 1 fungal endophyte in them. All the salt stressed plants had another fungal endophyte in them. And when we removed the fungal endophyte from those plants, they lost their ability to tolerate those stresses. When we put those fungi into the commercial varieties, whether it was their Dongjin or we were using a US strain as well, M206, then we were able to move that functionality back into the plants. We could do it in the wild plants as well. So the bottom line that I want to bring up is that now this is not uncommon. Breeding traits from native plants that are in stressful habitats is a common strategy. But the question is what are we breeding for? And in too many scenarios, we're breeding for for plant genes, or we're trying to engineer plant genes. We're trying to identify genes in the lab that confer heat tolerance and then move those genes into plants. Well, I guess what I'm here to say is that that's not enough. What we're doing is moving genomes. The stress tolerance in plants is a very complex phenomenon genetically. We know that you can move an entire organism into the plant and get these types of stress responses. Moving individual genes has not yet worked, and there may be a good reason for that. This is the way nature developed to do this, and that's the way BioEnsure works as well. So what I'd like to propose then is a new breeding strategy where we continue doing the same types of breeding that we're doing, but we add a new metric to it. And the metric is now symbiotic communication. We optimize the association between plants and fungal endophytes to increase benefits to their maximum levels. So I'll finish there. This is the strategy with which we've used to generate BioEnsure. We use a lot of criteria to analyze organisms to determine if they're worthwhile commercializing, if they can get through a regulatory process, if they do what we need them to do on a variety of crops. And BioEnsure now, we know, works on a variety of different plant systems, including cucurbits, tomatoes, and rice, corn, wheat, soybean. And we are now expanding. We've expanded our field trials tremendously. This year we have 30 field trials in 8 states in the US on 5 different crops. So with that, I thank you for your time, and I'd be happy to answer any questions.